So I was given the title to look at, can a farm really work successfully in a closed organic system? And now, so this was presenting to a bunch of organic growers and people involved in the organic industry. So it's a bit of a utopian dream. And I then spoke to the crowd and asked if I could change the title of the presentation to, if I can get that, what can we do to work towards a successful closed organic system? And that's in reference to, is it possible? I think technically, I think it is possible to do that, but very difficult in terms of you would have to be there with everything working to its full degree to be able to even get close to achieving that. But for me, and in reference to particularly the southwest, a lot of the southwest soils in the sort of the Cape to Cape region and the Blackwood area, a lot of the constraints with the soils, and we've got very old soils here. For me, it's all about information and making getting the right decisions, getting the right information so you can make the right decisions. And what I'll go through and show is how having the wrong information you can make poor decisions. So getting back to information, it is all about information, and a lot of people have probably seen this three-way Venn diagram with the right at the centre with is your ultimate soil health, and really that's to be able to get there. If you're in the centre, you're pretty much a closed system. So that's having every chemical, physical, and biological aspect working together, because each of these affects each other, and it's not one or the other. For the work that I do, it's about establishing the most limiting factor, and to do that initially is through soil testing. And I mean, I've been testing soil for the best part of 25 years. I've used lots of different labs and lots of read a lot, and the theories behind what the numbers mean can be a bit of a contentious issue, but what I'll show here today, it's really about the important stuff and making the right decisions, and you can only do that with the right information. So I'll then have a quick look at the important aspects, and we'll have a look at a case study of where I've got a number of years of data to prove up some of the theories we're talking about. So for me, going back about it was probably the mid to late 90s, I read Neil Kinsey's hands-on agronomy book, which introduced me to the Albrecht the notion of the balancing of the soil in relation to cation balancing on cations present on the soil colloid. Now, we were using laboratories back then that were providing this information, and back then it didn't seem to work, and a lot of people would say, well, it doesn't work for West Australian soils, it was made in America and it's only suitable for those conditions. And not that a lot of people gave up on it, but it was, for me, it was about trying to make the numbers work. So it really is which numbers to believe. So I started using a laboratory about 10 years ago, which I'm still using to this day. And about 5 years ago, to prove a point, which I'm about to show you some of the data now, I performed a lab off where I got five sites with 5 different land use. Got 5 identical samples from those sites and sent them to 5 laboratories. 4 of them, the most populous labs in Australia, and 1 of them in America. And it was to show the notion of how getting the right numbers can make a really big difference. So if you can see this, if you can go, this diagram here is just basically showing you cation exchange and how it works for a low CEC soil, which is a sand, to a higher CEC soil, which is more of a loam or a clay soil. So you've got your 4 main cations: calcium, magnesium, potassium, sodium, and most importantly, hydrogen. Now, hydrogen is basically through the plant roots, through plant growth. The plant wants to take up something that it needs for its growth, so calcium or potassium. It does that in exchange for hydrogen or protons, leaving behind the hydrogen in equal molarity. Now, the measurement of that hydrogen is a bit of a contentious issue, and which I'm about to show. So here we go. Here is the lab off. So I'll have a quick look at I don't want to bore people with lots of numbers, but what I'm trying to show here is on the left-hand side here. This is just a look at the cation exchange numbers with the different labs. So I haven't, because some of this information is controversial to some people, I don't want to be seen as bagging other laboratories, nor naming the laboratory that I'm using. If people are wanting to further that information, feel free to have a one-on-one with me, and I can elaborate a bit further. But so I've just labelled them labs A to E. The numbers on the left here are the numbers that come from the machines in the lab. So basically what's spat out by the machines. So we have calcium, magnesium, potassium, sodium, and hydrogen. The lab D is the American lab. All the other ones are the Australian labs. The one in yellow, lab E, is the lab that I use. Now, what I want to try and show to you is the numbers. So if we look at calcium here, the numbers that actually come out of the machine are within a laboratory error for different machines from different labs. But essentially, all of these tests that are done by these labs are the same methodologies. All the numbers for pH, for phosphorus, for potassium, for just about 9 out of 10 of the numbers come back within an accepted degree of error. The numbers on the right are the same as the numbers on the left, but are the percentages. They're converted just into the percentage of what is present on the colloid, meaning the clay or the humic material in the soil. So as you can see, if we look at calcium, they're pretty close. When you're converting them into percentages, there's a much bigger variation from 87% to 63%. If you go through these numbers for the magnesium, potassium, sodium, they're all within range of being pretty close. What is the big change is this measurement of exchangeable hydrogen. Its magnitude's different. So here, even with a pH of 6.7, where you would expect there to be not a lot of hydrogen, you can see the variance is huge. All the 3 out of the 5 labs are virtually 0. The lab E is a magnitude of, what is that, 20 times higher. So when you convert that into a percentage, the hydrogen ends up being a hell of a lot bigger than everything else. Here they're saying they're basically 0. The American lab is saying there's about nearly 5%. Here we are 26%. So proportionally, this changes all of these numbers, and you've got to make your decisions on these numbers. Now, for me, the importance of calcium and magnesium, as Kinsey and Albrecht went through, it's basically controls a lot of things like soil structure, availability of nutrients, and which flow on into biological constraints. So the better structure flows into having better biology and better nutrients exchange. So if I just have a look at, we'll go look at a couple more of these. Here's a site where the pH is a little bit lower. Again, if you were to browse through all of these numbers, they're basically the same. It's not until you come down to the hydrogen, we've got a magnitude of 20 again, or thereabouts. So the difference is when you convert it into the percentages again, these three labs here are saying, well, you've actually got far too much calcium. Got too much magnesium, whereas the real number, calcium is about half of this, and you actually need a fair bit of lime, and you need a fair bit of dolomite and a lot of potassium to improve this soil. The effect gets even more pronounced when the pH is lower. So here we have a pH of about 5.4, which is not dissimilar to a lot of soils around this area. This is actually a soil in Manjimup, really nice soil. It's probably not the red curry loam country, but it's a very nice soil that you grow just about anything in. Here again, we've got 4 of these labs showing these super low even numbers, but when we look at the hydrogen number, we've got a number of 10 here to virtually 0. Converted into percentages, again, these ones are saying you've got enough calcium, a little bit lower. The American lab is certainly better. Magnesium super low here. When you convert it to the real numbers, we've got a number of 85% hydrogen. So you can see here, if you use lab A, and you're basing your liming recommendations on this and not pH, you're going to make some pretty poor decisions, and it's just not a good use of resources. That's another one. It's basically showing the same thing. This soil had actually been worked and is almost pretty perfectly balanced, but it's showing the same trends. And what I'll show now, I'll just have a quick look at. A lot of people will ask, well, how do you know that these numbers are correct? I've been fortunate enough to work with Cullen Wines for the last 7 or 8 years, and I thought I'd use this as a case study because they're probably the farm is probably the closest closed loop system that I know of that's commercially operating and doing a lot of good things, and it's a credit to their commitment to doing what they're doing. They've been certified organic for 22 years. They've been carbon neutral since 2007. What I'm about to have a quick look at now is we've been testing 10 sites. This is showing we've got another year of data to add to this, but this is 6 years of data. And what they are doing is really trying to close the loop. Their biodynamic applications we've added lime and dolomite to order, some custom organic mineral applications. They make their own compost, and probably one of the most important aspects is an intensive cover crop program. We were graphing some of the data this time last year, and started seeing some really good trends. This is one's looking at soil organic, soil carbon levels. Now, when we started, this is not that the soils were that out of shape. They've been quite productive for a long time, but it's 40, nearly 50-year-old vineyard. decision was made to really fine-tune things. So when we saw this graph, we started where what we've shown is we've doubled, if not trebled, soil carbon levels over this period of 6 years. And every year they will pay a fee, a substantial fee to have carbon neutral status. These numbers were given to their auditor that audits their business every year, looking at everything from machinery passes to electricity to everything with a fine-tooth comb. They looked at the numbers and verified that the increases were real, and just to crunch the numbers down, it boils down to about nearly 2,000 tonnes of CO2 per year upon year, which is about 61 tonnes of CO2 sequestered per hectare per year, which is pretty significant. So what this means is that they don't get cash in hand for that, but they were able to gain credits so as not to have to pay the fee for the neutral status, which I believe they'll still continue because that goes into tree planting and lots of things. So these numbers basically added to a 2% average over these 10 sites, with about 4% increase in organic carbon year upon year. So during this time, we this is a look at all the other numbers. So this is available calcium, which is this exact same graph as exchangeable calcium. You can see there's the same trends across all the sites have gone through. So if I go through these fairly quickly, a quick note on calcium, why the calcium is so important, and this applies to every plant, whether it's a grass or grapevine or an apple tree. Calcium is required in such large quantities in every plant, and when you get your head around that it can only move one way in the plant from the roots upwards, so only through the xylem, whereas just about every other mineral can go up and down and be taken from older shoots to younger shoots. Not so for calcium. So you've got to have a constant supply of calcium into the plant if we can use the pointer here, I think that's working. All the cell wall and every cell wall in every plant, its structure is given by calcium. So if that's not in constant supply, the plant's going to use more energy to extract what it needs from the soil at expense of yield or resistance to pest and disease. So again, going back to the numbers, if you've got a false look at the number saying that you've got enough calcium, whereas actually you need a lot, you're not making the right decisions. Another one, look at magnesium, same thing. You can see we've started from, they weren't super low, but they've increased to damn near perfect numbers up here. Magnesium is critically important, not just for soil structure, but also for many plant processes. As you know, it's part of the chlorophyll molecule. It's the center of the chlorophyll molecule. And when I was putting this presentation together, I found this fascinating fun fact that it's basically the exact same molecule as the hemoglobin molecule found in human blood, with the only difference being iron at the center. And when you think about it, this is a porphyrin ring, which is common in some forms of biochemistry. But when your blood's low in iron, or if you're low in iron, you feel lethargic, and it's the same thing. If there's not enough magnesium, you're not making enough use of the sun, and you're not maximizing photosynthesis. So having that magnesium correct at the good numbers is where you need to be. Same thing for potassium, all good trends. Phosphorus, this was the start. We've actually had a decline with no phosphorus additions, with only, now phosphorus is super important for every single plant, more so in a biologically driven system. It's more important, or for this situation with a vineyard, we've got lots of really good background total phosphorus numbers. We don't actually need to add much phosphorus at all, but it's super critical for all biological interactions. It being the center of the the backbone of the DNA molecule. This blue thing here is all phosphate. So this molecule is in every cell of every plant, every cell of every bacteria, nematode, protozoa, everything in the soil. So if you have phosphorus limiting in the soil, it's going to limit your biological action just from the sheer fact of non being around. And there's more and more research that's showing that the lot of soil microbes, including phosphorus solubilizers and mycorrhizae, can detect when there's too much synthetic phosphorus in the ground and actually stop working. It doesn't kill them, but it stops them functioning, which then has flow-on effects through other layers of biology in the soil. So it's super important. And interestingly, one of the amino acids is called guanine, which is where guano gets its name from. There's boron, so I've been working on traces. And this is not necessarily adding a lot of boron. This is realizing it from the soil by improving organic carbon levels and biological activity. Now, this is an interesting side, which, going back to, some people ask, well, how do you know the numbers you're using from your lab are real and are working? This is a look at the exchangeable hydrogen and the decrease in it over this testing period, where they're basically in a range of being pretty close to ideal. Now, this is following 1, 2, 3, 4, even 5 years of sometimes significant amounts of lime and dolomite to balance the calcium and magnesium in the soil. And this next slide shows that, to me, it proves that the numbers are working, because you're not adding calcium or magnesium based on pH, but you're adding it based on calcium and magnesium, where across this time we've seen virtually no change in pH. There's a very slight trend up, but given the quantities that were added, this just proves, because the pH was virtually ideal before we started, and now it's even better, but with only marginal shifts up. From this work, they've certainly noticed increases in yield and quality. Increase, probably more importantly, relating back to the increase in organic matter, is that for every % you lift organic matter, or organic carbon, you're potentially increasing the water holding capacity of between 350 and 700,000 litres per hectare, which in a drying climate is significant. And probably more importantly, in relation to a closed system, it's they're reducing the need for any fertiliser additions and are becoming more and more in-house as time goes by. Now, a big part of improving soil qualities, more specifically increasing soil carbon, and which then has a lot of flow-on effects to enhancing biology, is the use of cover crops and multi-species cover crops, which we've just started doing in this season. We're still blending seed as we speak. And this is, in a regenerative agricultural sense, probably being seen as one of the most critical tools to go towards this a lower input system to get the most out of the soil and increase the soil health benefits from your topsoil. So it's essentially getting the right mix of a variety of plants that, as you can see here, this is a winter crop which got a variety of legumes and grasses and brassicas, and I believe this is being, this is going to certainly get a lot more popular, and is the science is slowly catching up with how it works, because we've really seen some and are seeing some great things come from this sort of work. So this is a vineyard situation where multi-species crop, and it's really got about getting as much biomass as possible. And I think the critical thing is to work out when to drop the crop, so as to not rob Peter to pay Paul, if you like. I think you can let the crop go too far and lignify too far, which can have some positive things occur, but in a perennial situation an annual situation like this, you don't want to let it go too far. So there's go back a slide. Here we see the Syrah rye really getting up there, and it's just a super thick crop. Generally, we want to be dropping it at around flowering of most of the grasses. Now, I think this is just to show, by balancing your soil and doing the right things in terms of cation balancing, you set yourself up for the foundation of creating soil structure via plants. So this is what the cover crops are doing. You're getting root biomass. Biology is being enhanced through this process, because you're creating this type of environment, which is what they want. So what this is demonstrating is, when you've got this situation, you're going to take advantage of the natural cycles. And just to look at this when a high pressure system comes over the land, it's, as it suggests, it's heavier pressure, it's higher pressure, so the air is heavier and is actually pushing into the ground. So if you haven't got this sort of situation in the soil, you're not getting enough from your biology, which these organisms like these diazotrophs, which are free fix bacteria that fix atmospheric nitrogen directly from the atmosphere. If you've got compaction layer up to here, you're not going to get any of this action of the oxygen and nitrogen rich air pushing deep into the soil. And then when a low pressure system comes over the land, the soil basically breathes out, releases CO2, and everything happens. So Here's one of the daikon tillage radishes that's seems to be a bit of a competition these days in the vineyard game. And this next slide will show that the guys at Voyager are probably winning that by a country mile, I would say. It's a very impressive plant, and this just seems to do really good things to the soil in terms of breaking up hard pans, and its residue really does good things to opening up the soil and encouraging lots of processes to occur. This is just another picture of a multi-species crop that's been left to really dry off as to get more of a mulching effect out of it. And I did this. This is actually my family vineyard. I did this to really keep a lot of carryover seed, but this seemed to work really well just as a mulch, as opposed to slashing it at this stage where you would have lost a lot of that. You gain some nutrition cycling, but here you're getting more of a mulching effect. Even working into summer crops to keep driving photosynthesis into the soil for soil building processes. So here, common summer crops which seem to work pretty well in the area of sunflowers, cowpea, lab, buckwheat and millet, they all do very well. A couple of shots there. Sunflowers are always a good look. And that's just showing that neighbouring paddocks completely bone dry. Bare soil is, sort of an inactive soil. Here we've got a green plant growing through the summer, continually photosynthesizing, increasing carbon levels, keeping the biology alive, ready for the following season. Moving into more closed system tools, making your own compost is a huge part of that, and going as far as testing, really pushing the limits to make the best possible product, as opposed to making a usable product out of waste material is the big difference to getting some worth out of, which is quite it's quite a lot of effort to take it to this sort of level, but I think it's hugely worth it, because it's really just an inoculation, a biological inoculation for the ground to really drive soil health. Been focusing on this element, cobalt, for the last number of years, where looking at the connection, but again, a biological connection with just about every single living thing on planet Earth needs vitamin B12 to function. It's the center of the cobalt is the center of the B12 molecule. So, and been looking at soil testing numbers for decades now. If you can see this number here on the bottom level here, as a typical cobalt number for a lot of the lighter soils in the immediate region. This 0.01 parts per million. It's just a typical number, and it's basically a trace of what it needs to be. And we've known this for years, which is why we supply supplements to animals. But when you think about all biological, microbial, and every biological organism in the soil is reliant on this pathway, if you like. So if it's super limiting, and bacteria are just like little cow and sheep. So if this is limiting, your biology is going to limit just as a little bit along the same lines as phosphorus and the DNA connection. If these are limiting, you're not making it easy for your biology if you want to get the most out of it. And just as an anecdote, I have noticed this number up here, which is a different soil, is 312 times this number. Now, this is a fairly common number that I've seen at least half a dozen times in some soils that I've tested around the place, and it's that scarpy country. This soil is actually off Pinjarra, where you're going from the flat levee plains up to the scarp, and it's typical through the Nannup and lots of parts of the hilly scarpy country through the southwest, where the cobalt it's on country that you could never spread any fertilizer or lime, or for that matter. It's inherently fertile component of these mineral rich soils that have super low other minerals like phosphorus, for example, but my theory being is that the cobalt's really driving this biological activity, that all of that mineral is tied up in their biological biomass, and you've got naturally fertile soils. So and we're certainly seeing some improvements in the soil by driving the cobalt in both soil and liquid applied materials. Do we want a question break now? I'll have it after this next slide. Now, interestingly, the single most cobalt or B12 rich natural material is cow manure, far above any other manures. So again, another theory, I think this is what Steiner was trying to tell us to do, is just to broadcast cobalt across the place to improve the biology of the soil. Happy to pause for a break now, Kate, if everyone can hear me still.
Here from the LCDC. Kate's microphone seems to have decided to play up, so I'm jumping in as a last-minute MC.
So I can hear you. That's great.
You can hear me? That's great. So yeah, if anyone has any questions, just type them in the chat box there, and I can read them out. We did have a question, Anthony, about sunflower seeding from Lisa. Do you have any advice about when and how would you seed sunflowers? Any particular method and time of year?
At the right time of year, well, a lot of those crops that I showed there, some of them were sown in late October or even early November. So it's a real opportunistic crop that we've done it twice. We've drilled them into some scarified ground, or we've also spread them and just scarified them after that. But yeah, they really only, as long as the soil's nice and warm, they really only need one good rain on them, and they're up and away. They're an amazing plant. They're like a mini drill rig on some hard ground, and got an amazing ability to survive.
That's great. Thanks, Anthony.
What method of seeding? Yeah, well, so either just drilling into a nice seed bed, depending on your gear. Like if you've got a disc drilling straight into some pasture, that's going to work okay. But otherwise, you might have to rough it up a bit. But it's you got to get them in the ground, because, as you know, they're a part of a bird seed mix, so the birds do love them.
Okay, we had a question from Crispin as well about was soil carbon level the main contributor to increased water holding at the Cullens?
Was soil carbon well? It's a perceived thing at the same time. That's just data from saying that for every % increase, you've got the ability to hold on to that much more water per hectare. So a lot of that is interacting obviously with your soil structure, but certainly, seeing more. I mean, they're a dry-grown vineyard, so we saw some really amazing resilience. This is the driest summer since a lot of people can remember, just gone, and I think that really paid dividends in this year. So I think yes, it would be. So there's lots of other things happening, like increased pathways from the cover crop biomass below ground as well. But that's it's a moving beast, the labile carbon.
Okay, if there's no other questions for the time being, back to you, Anthony. I think we'll probably have some time at the end for a few more as well, hopefully.
Yeah, sure. Well, I think I've just got some pretty pictures to go. Okay, well, I thought I'd put this slide in that just to say, just to really try and drive home that I think no matter what we're doing, is that the biology is going to trump regardless of what situation you're in. So if you've got a poor soil that's lacking mineral diversity and density, you're going to have a situation where your biology is not going to be balanced, and they're not going to be working for you, as opposed to when you get a mineral balance up and running, the biology will follow. I mean, it's not to say that applications like compost and compost extracts are not good additions. I think they're really good tools, but I think the sheer fact is that biology is there. It's about stimulating and getting them to work, and this is where I think the combination of the mineral balancing as well as cover cropping encourages the biology to do what they do, and to think that we're actually in control. I think got another thing coming, because some of the statistics here are just mind-blowing in terms of 5,000 species in 1 cubic meter of air, a kilometre up in the atmospheres, in that we've only identified as little as 0.001%. So I think they are by far in control. The next couple of slides, I was fortunate enough to win a government scholarship, agricultural scholarship, and it took me to Italy, where I saw this amazing farm, and talking about a closed loop system. This was pretty much as closed as you could get, in that here we have a custom designed biodigesting unit that was taking 100% of their waste from the farm. So this was a 1,000 hectare organic certified farm in southern Italy that had a range of crops. They grew a lot of cereal crops, from ancient grains to they were even growing some hemp, linseed, you name it, they were growing it. Significant olive groves, significant vineyards, all sorts of things. So they had a system in place where they were every single bit of organic waste material, apart from the prunings from the heavy woody stuff they weren't putting in, but everything else was going into this system where they had these massive bays here. So all the grape mark, all the olive pomace, and everything else. So pretty significant operation that would then feed into these enormous digesters, which was going back at the end there. So they were auto feed, where everything would go in, digested. They're producing methane, which is supplying 100% of their electricity requirements. But it also had this auto outfeed that the spent digested organic material was supplying 100% of their fertilizer requirements. So it was about as closed as you could get. So it was pretty impressive, to the point that this is a picture of a seed decorticator that was just been installed pretty much the month before I visited them, where they were any bean, grain, lentils, cereal, whatever you can imagine, was being cleaned and bagged. Here, this is the outside of this unit where they were collecting all the husks, and that would go into the digester. So they weren't letting anything go to waste. And then they had another facility where they were making their own flour and pasta and you name it on site. It was pretty impressive. And where are we now? So this is about as closed loop as you could get, and you know, they were on some pretty amazing soils and an amazing climate that allowed them to do things. Quite mineral rich ground, but a pretty amazing setup. So I'll wrap it up just a bit. This is some of the things that I'm doing. We've just been supplying these organic biostimulants and whatnot for the last 9-10 years. More recently, last year, we installed this mini batching plant where we're now doing custom blends of any fertilizer. So we take it from paddock to plate sort of thing, where we'll test the soil and do prescription blends to suit each site. And we have a full range. People can make their own blends if they like. So full range of these sort of materials like guano and humates, all the trace elements, all inoculants, compost extracts, and full range of biostimulants, which are proving to be an invaluable addition to anyone's program. And lastly, I thought doing some more research on some microbes. This is an interesting one. It's called the Demodex mite, and at the age of 18, I think it is, 100% of people test positive to this mite, which lives in the pores on your face. Now, it's believed that they feed off the oils and things that are inside your pores. They're blind, and apparently they don't have an anus. So when they fill up, they explode all over your face. So I just thought that would be a good thing to finish with, just saying again that I think biology is in control. So, but they're doing something good for us. So I think if you can hear me, Joe, I think that's comes to the end of that. If anyone's got any more questions.
Anthony. And yeah, thanks for taking the time to share your expertise with us. So if anyone has any questions, we've got a few minutes, so just type them in the chat box. Yeah, we had one from Watson and Laura about the Italian farm.
Yeah, that was right in southern Italy. The town, it was just outside of Brindisi. I think that's how you pronounce it, Brindisi or Brindisi. Believed possibly to have some mafia connections. It's where they grow a lot of the tomatoes in right on the southern end of the boot.
And another question from Leonard Russell: can compost tea be used instead of compost?
Look, it depends on what you're wanting to do with it. Look, I think compost tea is an interesting one. I think, I don't know if anyone's been listening to some of John Kemp's podcasts. He recently interviewed Ray Archikera on it, who's a well-known consultant in America, and he had some interesting things to say in that. And I sort of feel the same way in that, look, I've made compost tea for 20 years, and I think it's a good tool. I think, as this guy said, I think for about 10 years there, the world's been hoodwinked thinking it was the solution to everyone's problems, which it just plainly isn't. But to answer your question, Lenny, it can be used as an inoculation, but I think a compost extract is better. This idea of brewing microbes, which has been around for a long time, and growing them in like a 24-hour process, is initially was done for foliar protection and in things like vineyards and orchards, which I personally haven't seen it control things like some of the fungal pathogens like powdery mildew. Compost extracts, cold extracts, where you can do them in half an hour, a little bit more of a shotgun approach, I think are a much better tool as an inoculation. But to get the real soil conditioning effect that you see from a compost application, I don't think it can replace it. I think it's a good tool. So if you're doing it regularly in a pass of soil drenches, instead of using carrier water, you're using a compost extract, I think that's great. But to rely on it as a cure-all is not the go.
Okay, the next question from Kate: is it a bit of a waste putting on biostimulants unless you have remineralized your paddocks first?
Certainly not a waste, I don't think. It depends on where you're at with the mineralization process. So I think what I was trying to get across before with the biology, there is truckloads of biology in every soil, even if it's been under a nuclear program of chemical abuse. It's pretty hard to get rid of it. The encouraging natural processes to get the biology working, biostimulants are certainly a good inclusion and not a waste of time. But the remineralization is certainly important. And look, it's just the fact that a lot of the soils, particularly where we are on the Cape to Cape, they're geriatric soils. They're so old and low in minerals for the most part. I mean, we've got some beautiful soils in like the Williabra Valley and where they're naturally fertile, but where we're a lot of country out in plateau country out north and south and east and west of the local area, pretty lean soils. So you just got to get the minerals in there. They don't arrive from the ether, unfortunately.
Okay, so if there's no other questions, I'd like to say a big thanks again, Anthony, for that really interesting and informative presentation. Thanks so much for your time this afternoon.
No problem. I'm glad we got around some of the audio issues.
Yeah, it can be a little bit tricky sometimes, but I think we got there. And lastly, many thanks to you, the audience, for joining us for this afternoon, and apologies for the audio issues that we did have. Your feedback is really important to us. So if I can ask you to complete our webinar evaluation form before you sign out, that would be great. And hopefully Kate can bring that up now. But yeah, thanks again. And
No worries.
and yeah, thanks, Anthony, and thanks for joining us, everyone.
Okie dokie.